Quorum sensing is a mechanism that prokaryotes use to communicate with each other based on cell density. Quorum sensing often initiates group behaviors in cell colonies. Prokaryotic organisms sense population density by emitting chemicals called autoinducers that can be recognized either between cells of the same species or between different species. Microbial quorum sensing is similar to how a party works. An appropriate density of people must be reached before the party can really begin. It is only after enough people show up that people really begin to party. Cells constantly emit as well as receive signals to communicate with other cells. Because lots of different microbes are sending out signal molecules to other microbes, the extracellular environment contains certain levels of cell-specific autoinducers. The number of signal molecules is proportional to the amount of cells in the environment. If a cell receives the appropriate number of signals, it knows that there are enough cells of its own kind in the environment to initiate a group response. The group responses often involve gene activation or deactivation. Just like the fact that parties don't begin until a certain density of people has been reached, cells also wait until a certain cell density has been reached before initiating group behavior. Autoinducers trigger a pathway of biochemical reactions that modify gene expression, initiating group behaviors such as bioluminescence, biofilm production, virulence, and sporulation, among others. Many cell behaviors such as bioluminescence and biofilm production are not useful if only one cell is involved, since single cells can't produce enough light or biofilm to be of much use. However, many cells activated at the same time can have a noticeable effect on the environment. There are two different types of autoinducer molecules. Gram-negative bacteria use acylated homoserine lactones, whereas gram-positive bacteria use processed oligopeptides. Both signal molecules depend on membrane properties for dispersal from and entrance into the cell. Cell membranes composed of lipid bilayers are impermeable to proteins, but are permeable to small nonpolar molecules. So, in gram-negative bacteria, whose autoinducer is relatively small and nonpolar, the amount of autoinducer in the extracellular environment is the same as that in the intracellular environment. However, in gram-positive bacteria, autoinducer molecules accumulate in the extracellular environment and are bound to receptor membrane proteins rather than passing straight through the gram-positive membrane. The bacterium Vibrio fischeri provides the best understood model of quorum sensing for gram-negative bacteria. The Hawaiian bobtail squid utilizes the bioluminescence of Vibrio fischeri in order to conceal itself or to attract prey. Generally, gram-negative bacteria have a regulatory protein that produces autoinducers as well as another sensory regulatory protein to bind them, triggering a gene regulation response. However, some exceptions utilize a two autoinducer sensor pair for quorum sensing regulation of more than one gene. The soil bacterium Bacillus subtilis provides another model of quorum sensing for gram-negative bacteria. Bacillus subtilis uses quorum sensing to trigger spore formation to survive through harsh conditions. Gram-positive bacteria encode their autoinducer in their chromosome. Transcription and translation of the autoinducer gene produces a peptide that must be modified by cleavage. The cleaved peptide can be secreted by the ATP binding cassette transporter, which is just a membrane-bound transport protein, to transport the oligopeptides out of the cell. When an oligopeptide binds to a sensor kinase, which is another membrane-bound protein, a phosphorylation cascade occurs, resulting in upregulation of certain genes. So to summarize, quorum sensing is a mechanism that cells use to sense cell density. If the cell density is appropriate and environmental conditions are right, cells may initiate a group response. Microbes send out and sense information about cell density by both emitting and receiving signal molecules called autoinducers. Because the membrane components of gram-negative and gram-positive bacteria are pretty different, the two types of bacteria use different signal molecules. Gram-negative bacteria use an acylated homoserine lactone molecule as their autoinducer, while gram-positive bacteria use processed oligopeptides as their signal molecule. Some group behaviors that can be initiated are bioluminescence, biofilm production, virulence, and sporulation. We hope that this video has been helpful in understanding quorum sensing. Thanks for watching.